This is part 2 of the sickle cell allele slash malaria case study that we were looking at. Part 1 ended with an individual whose parents were carriers. This individual had a 50% chance of inheriting SBS, or the sickle cell allele hemoglobin, from mom and a 50% chance from dad. So the total probability of these parents creating a child with sickle cell anemia, in other words, their alleles for the beta subunit of hemoglobin, or the instructions, the ATGCs for this individual is a sickle hemoglobin allele and another sickle hemoglobin allele. So this individual has sickle cell anemia, and they are unlikely to survive long enough to reproduce. Or even if they do live long enough to survive and reproduce, we would expect over time that the HBS allele be weeded out of the human population. If this is the time in generations and this is the frequency of HBS, we would expect over time this allele to be filtered out of the population due to the fact that it does lower the reproductive success of individuals who do carry the allele. But there is an exception, and the exception is for populations that are exposed to the malaria parasite, or put another way, for populations where the malaria parasite is endemic. So we are going to now change this individual to a carrier. So this person is HBA, HBS. And what scientists noticed is that the frequency of this allele is much higher than predicted in regions that are endemic to malaria, and they wanted to see if there was a relationship between this allele and malaria resistance. And so what they did, and again, if you're interested in seeing the map, I don't have it pulled up, but you can always check out part 1 to see the map where if you superimpose the sickle cell allele frequency on top of regions that are where malaria is endemic, you see a striking pattern. And so what scientists did to see if this pattern was real or if it was just imaginary is they went to regions where the malaria parasite was endemic, and they looked in hospitals. So we have hospitals over here, and I guess that doesn't look like a hospital. We have hospitals over here, and outside of the hospital we just have people living in close proximity to the hospital. And scientists intentionally chose regions that were sort of plagued by the malaria parasite. This is my Anopheles mosquito, which carries the malaria parasite, because they wanted to compare the allele frequency of individuals in the hospital with malaria, compare those allele frequencies with the allele frequencies of individuals who were not in the hospital. And so in the data set that we looked at, the 500 people sampled in here who were all sick with malaria were categorized as cases. These people were categorized as control. So let's just think about this for a minute. And remember from part 1 we said that people with HBA had an A nucleotide. People with HBS, the A was swapped with a T. And so, for example, when we're looking in cases, we look at person number 1, and we sequence their DNA, and we specifically look at the gene for the beta subunit of hemoglobin, and we see that this individual is AA. What that means, let me draw the other A in a different color so that you can sort of get an idea about where these A's are coming from. Let's just say that this gene came from the person's mom, and the other gene, or I guess I should say the other allele, came from the individual's father. What this means is that this person number 1 is HBA, because both of the alleles that they inherited from each parent have the A letter instead of the T letter, and as a result, this individual is producing completely normal hemoglobin. And so we looked at the DNA sequence of 500 people from the hospital, so 1 through 500, and then we looked at 1 through 500 people in the control population, or in the control sample. And here's what the researchers were thinking. They were thinking that if there is a relationship between this allele and the malaria parasite that's carried by this Anopheles mosquito, and specifically if this allele is somehow providing some sort of benefit to people who have it, then the logic is that maybe people in the control population have more HBS alleles than people in the cases population. And maybe the whole reason that these people right here aren't in the hospital is because they carry the HBS allele, and that somehow gives them resistance to the malaria parasite. So that was the thinking, and then of course we can use statistical analysis to determine whether or not the different allele counts between cases and control is statistically significant.